Acids and bases are inorganic molecules that are essential in life. There is a misconception that acids have sour taste, while bases have a soapy feel. Remember that you should not, in any case, use your sense of smell, touch, or taste to determine if a substance is an acid or base. There are safer and accurate ways to do that. As mentioned earlier, the regular and cheaper way to determine if a substance is an acid or base is to use a litmus paper. When a substance is dissolved in water, the resulting solution causes the litmus paper to change color. Blue litmus paper turns red under acidic conditions, and red litmus paper turns blue under basic or alkaline conditions. In every sample, you need to use both a red and blue litmus paper. Why both? Why not either red or blue? Aside from acids and bases, we have substances that have neutral pH. We will discuss more on this later. If a red litmus paper in a neutral solution did not change color, we will immediately conclude that it is acidic, which is wrong. Placing a blue litmus paper will confirm the pH of the solution. In this case, if the blue litmus paper turned red, then it is acidic. But if it remained blue, then it is a neutral solution. The litmus paper can only determine if it's an acid and base, but pH has different degrees from strongly acidic to strongly basic. A more accurate way of determining pH is using a pH paper. This is determined by dipping part of the paper into a solution of interest and watching the color change. The packages that pH paper comes in often includes a color-coded scale indicating the pH that something has when the paper turns a certain color. For instance, if the paper turns a light greenish blue, the pH may likely be around 8 to 10. More accurate pH measurements are obtained with a pH meter. A pH measurement system consists of three parts: a pH measuring electrode, a reference electrode, and a high input impedance meter. The pH electrode can be thought of as a battery with a voltage that varies with the pH of the measured solution. It provides a quantitative value of the pH of a solution. What makes a solution acid or base? Based from the chart, HCl or hydrochloric acid is considered as the strongest acid. When you dissolve HCl in water, it will break into ions, specifically H plus or hydrogen ions and Cl minus or chlorine ions. Solutions with large number of H plus or hydrogen ions are considered acids. On the other end of the chart, NaOH or sodium hydroxide is considered as the most basic. When NaOH dissolves in water, it will produce the ions Na plus or sodium ions and OH minus or hydroxide ions. Solutions with large number of OH minus or hydroxide ions are considered basic. On the other hand, an equal number of H plus and OH minus is considered a neutral solution. What will happen if you mix HCl and NaOH? The acid and base react, forming water and salt. When a strong acid completely neutralizes a strong base, the pH of the salt solution will always be 7. This process is called neutralization. Take note, it is dangerous to mix acids and bases since some may produce violent reactions. In our lab experiments, solution A is baking soda, which is basic. When water, solution B, was added, there was no change in color. However, when vinegar, solution C, was added to solution A, the solution color changed into pink. In this case, the vinegar was able to neutralize but not completely the basicity of baking soda. You will learn more about acids and bases in your chemistry class. Based from the chart, we can conclude the following. pH of 7 is neutral, neither acidic or basic. pH values less than 7 are acidic, while those greater than 7 are basic or alkaline. The further the pH value is from 7, the stronger the acid or base. Thus, a pH of 1 is a very strong acid. A pH of 14 is a very strong base. Most life forms do best in environments with pH values from 6 to 8. Our bodies try to maintain the pH of our blood within this narrow range. The lungs control your body's pH balance by releasing carbon dioxide. Carbon dioxide is a slightly acidic compound. It's also a waste product produced by cells in the body as they use oxygen. The cells release it into your blood and is taken to your lungs. When you exhale, you're expelling that carbon dioxide, a process that also helps regulate your body's pH balance by reducing acidity. The amount of carbon dioxide you exhale is a function of how deeply you inhale or exhale. Your brain constantly monitors this in order to maintain the proper pH balance in your body. The kidneys help the lungs maintain acid-base balance by excreting acids or bases into the blood through different buffer systems. A buffer is a solution that can resist pH change upon the addition of acidic or basic components. It is able to neutralize small amounts of added acid or base, thus maintaining the pH of the solution relatively stable. The kidney's effect on acidity works much lower than that of the lungs. A blood pH imbalance can lead to two conditions: acidosis and alkalosis. Acidosis refers to having blood that's too acidic or a blood pH of less than 7.35. Alkalosis refers to having blood that's too basic or a blood pH of higher than 7.45. This figure shows what happens to your body during acidosis or alkalosis. Aside from maintaining the blood pH, we use acidic fluids such as saliva, sweat, vaginal fluids, and gastric juices to keep harmful organisms out of our bodies. To maintain the pH in our body, we should also consider the food we eat. Maintaining pH is also necessary in different areas. Soil is a complex system that involves many different factors that are affected by soil pH, such as microbial activity, fungal growth, availability of nutrients, and root growth. Under acidic conditions, many minerals in soil become soluble, releasing toxic metals such as aluminum. Some nutrients become less available at lower pH values. Under alkaline or basic conditions, the soil can become deficient in nutrients such as zinc, copper, iron, manganese, boron, and phosphorus. Water that has a pH that is too low or too high can be harmful to fish and other aquatic life. Water with pH below 5 can inhibit reproduction or lead to death, and young fish and other aquatic organisms are especially susceptible. Water with a pH below 6.5 can also inhibit growth. At high pH values such as pH 9, most ammonium ions are converted to ammonia, which is also toxic to fish. Whether in treating drinking water or wastewater, pH is important. The pH of drinking water should be between 6.5 and 8.5. Low pH drinking water can degrade pipes, causing toxic metals such as copper and lead to leach into the water supply. Water with a pH that is too high has an unpleasant taste, and the effectiveness of disinfectants such as chlorine is decreased. Swimming pools typically have pH values in the range of 7.2 to 7.8. If the pH is too high, the effectiveness of the chlorine disinfectant becomes too low, making the pool susceptible to algal growth and preventing it from effectively killing viruses and bacteria. If the pH is too low, the water becomes irritating to the eyes and nose, and it may corrode plaster or metal surfaces. In the food industry, pH is measured to test for quality, to control microbial activity, to control the taste and other properties, and to prolong the shelf life of food. For many foods, the pH must be kept within a narrow range so that the food can be conserved for a longer period. Similar to other processes that involve microbial activity, pH affects many different aspects of the beer brewing process. The pH of the wine must be kept at a low level to prevent bacteria from degrading the wine. Lower pH wines will tend to mature more slowly and will be less susceptible to spoilage. The pH of wine also affects its taste, as more acidic wines tend to be dry.